Good morning, everybody. We're just waiting for a few people to attend, and then we will start the webinar. Good morning, everybody, and welcome to this Whiteside-hosted webinar, where you'll all have the opportunity to learn more about the solutions that Swift Biosciences has for SARS-CoV-2 sequencing. So my name is Jock de Preer. I am the NGS product at Whitehead Scientific. And before we start, I'd like to just go through some housekeeping. First of all, you'll see that you'll all be muted. At the end of the presentation, there will be a question and answer session where we will unmute everybody. You also have the option in the interim during the presentation to make use of the chat box. So if you have any questions, please feel free to type your questions in there. Without further ado, I'd like to introduce Gerardus Dambruskas. He is the Director of Commercial Operations at Swift Biosciences, and he will be telling us more about a presentation titled SARS-CoV-2, Full Genome Coverage Powered by Swift Biosciences Amplicon Panels. So I will give the control to Gerardus, and we can take it from there. Gerardus, are you able to take control? Yes, okay.
Hello, everybody. I hope now you can hear me. I was on mute. Yeah, so hello, everybody. Jacques, thank you very much for the introduction. Gerardus Dambruskas, and yes, so I work with Whitehead Scientific, our distributor for South Africa. And today I'd like to introduce our SARS-CoV-2 panel for full genome coverage, powered by in my title, powered by Swift Biosciences Amplicon panels. So next slide. So I would like to start with the protocol itself, which is an elegant protocol. And as you can see here, it's a multiplex PCR to enrich the regions of your interest. So the way we designed the Amplicons is to cover without any gaps my multiplex PCR, the full SARS-CoV-2 genome. And then you go into the next step where you would be adding the adapters and indexing them. So there are 2 PCR steps indicated here, and then you obviously get the finished library. There is an optional step here, whether you want to normalize the libraries using the Normalize step from Swift, which I will come in the next slides. So Normalize would allow you to pool the libraries into equimolar concentrations. So you have an end pool at a specific concentration, and each library will be represented evenly. Now, if you look here, we have on the panel the 99%, almost 100% of bases covered. There are 345 amplicons with an average length of 150 bases. The kit includes starting from a cDNA, all the reagents are needed to complete the library, to complete the library ready for sequencing. From a third party, you would need the total RNA extraction and the cDNA synthesis. Everything else is included in the kit. Then we have an option to multiplex up to 384 combinatorial dual indexes or unique dual indexes. The recommended depth is 50,000 reads per library for the yes or no detection, whether you're looking for the positive or negative sample. We also will have, would recommend 1 million reads, although what we are seeing now that about 250,000 reads are sufficient for the variant calling. Well, it depends on the sample nature. It depends whether you're looking with whether you're working with the wastewater samples or whether you have the nasopharyngeal swab from a clinical sample. So that would depend whether you're looking for one specific variant or if there is a mixture of variant when I'm referring to the wastewater samples. That would probably be 1 million reads for wastewater samples in this case. Okay, so the next in the next slide, there is a lag in any time I press the button. Okay, there is so just to recap, what SNAP stands for, or we call our panels SNAP, which is an acronym for Swift Normalize Amplicon panel, and the normalization step is optional and it is included in the panel. What it makes stand out against other Amplicon panels that we developed, obviously the elegant protocol that comprises 2 PCR steps. Then the Amplicon libraries can be, the concentrations can be straight away measured by bioanalyzer or qubit. We recommended previously on our Amplicon panels to be quantified using the real-time PCR, qPCR. This now can be shortened, and bioanalyzer or tape station in your laboratory is enough. And then also, just to finish, the Normalize is included as an optional normalization step in the library preparation. Okay. So supported applications and samples. So SARS-CoV-2 kit, SNAP SARS-CoV-2 kit can be used for just a yes plus or minus detection. Then whether you want to confirm if it is a positive or negative sample, for epidemiological studies, for surveillance, public health surveillance studies, whether you're working with waste, water samples, soil samples. Variant calling, that's very important. And then all types of the upper respiratory tract samples that are indicated here. SARS-CoV-2 workflow. Just to recap, the cDNA upstream can be used as an input into the library, which would take 2 hours, the library prep itself, and the optional normalize step, which would be an extra hour indicated here. And then the library is ready for to be sequenced on Illumina sequencer. Now, needless to say that we are working specifically on Illumina sequencers. There might be some developments on other platforms, but at the moment, this is not the option. And then to complete, we do have a command line, which is the set of the few steps that are connected in a seamless workflow, and we can share with you our developed piece of analysis with your bioinformatics. The question is that you have to have the bioinformatics be able to help you understand the command line that we developed at Swift. And I will come to this in my next slides. Just to recap, not just we have SARS-CoV-2 covering the full genome, we have the S gene, just specifically S gene, and I'll come and show in the next slide, that is 100% covered with smaller sizes, with smaller number of amplicons. That allows more sample pooling compared to the full SARS-CoV-2 panel that we have. And I won't go into the details here, as I already showed. And just to recap, there's a 2-step protocol, and the 3rd step would be the optional normalize. You can also pool the libraries based on your own method of pooling, but normalize is there, so you can take advantage and use normalization step. So should be the next slide should be S gene, SARS-CoV-2 S gene coverage. So here's the IGV shot of the S gene indicated here, and you can see here the reads that have very good coverage across the S gene. So the blue bars here are the actual represent the amplicons, and you can see here there is an overlap, a really nice overlap of the amplicons. And here is the dot plot, and there should be dots here. I don't understand why they're not presented. So between the red and the blue line, I'll come back to the slide. There should be dots that are scattered. So these, each dot will represent the amplicon, and you can see the mean coverage of that region in the blue, which represents the 5x mean, and the red represents the 20% of the mean. And these are the set parameters from our bioinformatics, and the amplicons should be scattered in that region. And it's not showing, and I apologize, I don't understand my original slide it was. So we use the synthetic DNA that we will assay each of our panel before the panel gets released into the production. So then we are absolutely sure that each amplicon is falling within these thresholds. So that's how we will QC every new panel and every new batch. So we have a really good composition of the amplicons that would give you this coverage. So we are doing this by shallow sequencing in-house before we are releasing any panels. Next slide. So this is the Upstream Kit that we'd recommend. It's from the superscript 4 from Thermo Fisher. And this basically enables you to synthesize the first strand cDNA. We don't recommend synthesizing the second strand DNA. The reason being that it is an extra time and taking extra reagents. So one cDNA would be enough for the primer to bind, and in the next cycle it will create the template for the other primer to bind. Therefore, one single-stranded cDNA as an input is enough. Now, going into the normalization step. After the library preparation, you can use your own method, pulling the samples, or you can use normalization step that's been built into the kit. So this is your index libraries, which are indicated here. So these are non-indexed libraries, and these are indexed libraries, which are part of the library building procedure. Then you would use the normalize one, which is the enzymatic step of normalization, indicated here, where normalize would mark a specific, a certain concentration of each library. This is a very schematic, very graphical approach, so to speak. Even those libraries are at different concentrations, the specific amount of the library will be marked with normalize one in a separate tubes. Then you will use one single pool, so pull all the 384 libraries, if you wish, into a single tube and go into the normalize 2, indicated here. What normalize 2 would do, it will just get rid of the excess of the unmarked concentration. So you will end up with the libraries that are normalized, or they are equimolarly pooled in the same pool, ready to be sequenced. So that's an elegant step, and we routinely have the customers already successfully using normalization step with our products, not just with SARS-CoV-2 panel. By the way, it was developed with our whole genome sequencing libraries that are being made by using our whole genome library Turbo or Sonic. Those two kits would seamlessly integrate normalization step. Okay, so these are the sequencing recommendations here. What I wanted to draw your attention that we recommend 2x150 parent sequencing, and that there is no Phi-X required in any of our Amplicon libraries, whether they are SARS-CoV-2 or whether they are gene-specific or disease-specific Amplicon panels. And then, as I said, I will repeat myself, 50,000 reads for the yes or no detection, and about 1 million reads for the variant calling. And this is the snapshot of different number of samples that you can pull using different chemistry, whether it is on a MiSeq or MiniSeq. This is all included in the in the workflow protocol. I won't spend more time on here. Now I wanted to just a little bit focus on the data analysis workflow, the steps that we have to get us all the way to the variant calling. So it starts with the adapter trimming. The adapters are the synthetic sequences, and then they will trim them, and then we trim anything, and we trim anything that are 30 bases long, less than 30 bases, and then we perform the alignment. We perform the alignment before we trim the primers. Okay, so First of all, you have to trim the adapters, and then you're still left with the primers that you used. So we'll need to get rid from both ends, from read 1 and read 2, the primers. And then so we developed the primer clip. So before we trim the primers, we do the alignment. Why we do that? We notice there's no loss in data, and secondly, this is computationally faster process. So after the alignment, we get the primer trimmed by the primer clip. This is developed by Swift, and we can share that script with you as well. Then we run the script for on-target and coverage metrics calculation, and then reporting. And reporting is that you could basically get in Excel spreadsheet the variants that have been identified versus the reference sample. And this is the open source tool. It's on a GitHub. It's also on our website. If you scroll to SARS-CoV-2 panel and look into bioinformatics files, we can share that data. And there's also demo data you can download and play with that open source command line that you see here. Okay, now the key performance across viral copy numbers. So this is something that we developed in our laboratory. We tested the panel, basically looking at the specificity of the panel first. What we did, we used a SARS-CoV-2 control synthetic that can be commercially purchased, and then we mixed it with 50 nanograms of universal human reference RNA. So as if to mimic the real scenario where the RNA is present in the human RNA. And then we made a synthesis, cDNA, and then we made the libraries and sequenced on the MySeq, indicated here, and downsampled. So we normalized it to 50,000 reads or 1 million reads. So on this graph here, you have the percentage of the reads aligned. This will give you the idea of what you would expect doing your own sequencing, depending on the number of copies you have. Whether you have 100 copies, 10 copies, 100, and so on. So the more viral input you have, the more reads would align to the genome, as it's shown here. And this is not impacted by the read depth. Okay, whether you're doing 1 million or 50,000 reads, you still more reads would map, and these lines would coincide or overlap. So this is your specificity. Okay, and this is how it works. On the right hand, you see the percentage of the bases that have the 10x coverage or more, or more. So if you have, even if you have 10 copies of the viral genome with 50,000 reads, 10 copies with 50,000 reads, you have 20% of bases that have more than 10x coverage. That's pretty good. So it's even with 10 copies. Now when you have 100 copies, that goes to 80% of the bases that have more than 10x coverage with 50,000 reads. That's pretty good. But there's a complete different story when you have 1 million reads. You basically have 100% of bases that have a 10x coverage. So that's your sensitivity. It's just to give you some sense of an idea about how sensitive the assay is. Even with 10 copies, having 1 million reads gives you 80% of coverage, which is pretty good. Okay, next slide. Now this is, we are looking into the what has been published in one of the flagship papers that we recently had with our panel referenced on it, or not referenced, but it was used in the study. So on the this axis here, you can actually see the coverage uniformity. This is the qRT-PCR CT values here, and this is the coverage. This is the mean depth, and as you can see here is that the mean depth, and these are the samples, so each bar is the sample, and this is the publication from Arizona State University, just 2021. I think it's even published in February, on the 10th of February, something like that, and there's 91 samples from wastewater from 11 states. So this is what I remember. And these are wastewater samples. So these are the collection of the potentially few viral strains that the researchers were identifying. So the purpose of why I'm showing this is that they said there is no correlation of the SARS-CoV-2, the coverage uniformity of the samples versus the qRT-PCR CT value. So on this graph here are the CT values. So if you go here, so samples have 35 CT, and you have not really that much difference between the coverage and the CT, except here where they had few samples that there was a correlation. So there was a lower CT values, and they had a lower, higher CT values with the lower viral load, and they had a lower coverage indicated here. But then it came back, so there's no really a correlation. The correlation, what I noticed comes from the, so going back, why there was no correlation. The customers or the scientists in the paper, they discussed that RNA gets degraded much faster in the wastewater samples than in the clinical samples. Therefore, you don't see that really no much correlation, but there is a correlation between the CT value. So the higher the CT value, the lower the viral load, the lower the coverage in the clinical sample. And this is what was important to highlight. So then we look at the complete SARS-CoV-2 genome coverage, experimental work done in our lab. This is the IGV shot of our NGS reads indicated here across the genome, shown here. As you can see, the coverage is pretty uniform, and there are no gaps indicated here. And these are the two iterations of the genome. The first one was 98% coverage, and you can see here we had few gaps. And then with the second iteration, which is the latest release of our panel, which says additional coverage, we have virtually every single gap covered, and it comes to 99.7% genome coverage. And by the way, this is sequenced with 100,000 reads per sample. So that's how you see the coverage here, shown here. Right. So going back to this wastewater samples from our Arizona publication. This is the viral genome, and then on the y-axis you see all the sample coverage across the viral genome in all 91 samples. Each sample marked with cyan had the coverage in those regions. So this is the across the viral genome. So all the viral regions are covered. You can actually see. Now asterisks here is shown where the coverage was down to 1x. But bear in mind, these are wastewater samples with a high degradation of the viral RNA. And but still, as you can see here, with the exception of those, we still have a pretty good coverage across the across viral genome in each of the sample. The samples that are marked in red, those regions were covered with more than 90% coverage. Okay, and they had out of 91, 52 samples that had more than 90% coverage, which is pretty good, given that there is a highly degraded samples. Okay, so in this slide, I wanted to show, it demonstrates the number of SNVs, single nucleotide variants, or the variants that were called, and how they correlate with the mean depth. All right, so as you can see here, that most of the variants were called with less than 500x coverage. And then on the right, you see the important findings, and I just selected this from the paper. They were able to find the D to G mutant, which everybody knows about it now. That's been found in the variants across those countries indicated here, including South Africa. What is important that they found the D2V variant in a very low frequency in the wastewater, and it's in a spike protein again. And yeah, that just the new variant that was reported in their paper. I just wanted to draw your attention. The next slide. Okay, so what I did here, from the paper, I selected the snapshot of the S protein, or the S gene, spike gene, indicated here. And then on the y-axis, you see the number of samples that had these mutations identified. And they are like six samples probably here had this mutation localized on the S gene. This was quite interesting. So positions with multiple variants, so multiple variant, were marked in those in the gray dots, and then also in the findings that they, as I said, they found D2G mutation, D2V mutation, and then 263 variants were identified in wastewater samples that were not reported in the clinical samples as of November 20th. What that tells us that wastewater is a really good tool to evaluate the epidemiological status, or the good surveillance tool to identify the mutants present in the area. Interestingly, as you see, there's a lot of mutations that found in S gene, but a number of those mutations are synonymous substitutions or nonsense mutations. So they don't change the actual sequence of the spike protein, as you would expect. And this is actually good news, of course. Just to show how this mutation is being covered by our Amplicons. So here's the IGV shot of the mutation location on the actual sequence, and these blue lines indicated here are the amplicons. So at least one amplicon covers the mutation. So there's a part of another amplicon here. So talking about the D2G1, D2G mutation, very early last year, we tested our panel at the New York University Grossman School of Medicine. And Adriana was our beta tester. And she ran the samples. These are clinical nasopharyngeal 29 swabs. They evaluated the qPCR, and the CT values ranged from 16 to 42 cycles. And all I wanted to show here that she managed to detect that mutation in almost all her 29 nasopharyngeal swabs. It was just a technical paper we published together with Adriana. And in the next slide, I wanted to show the phylogeny of the D mutant, D variant, I should say, and then the mutant into the G. And this is the across this is the timeline, and this is the phylogeny, and how it spread then into G. The G variant, how it spread across the world. This is the screenshot of the nextstrain.org. So this is a historical already fact. In the next slide, I wanted also to highlight how our Swift Amplicon technology works in case there is a mutation in the primer binding site and in case there is a mini amplicon that can be generated. So all just stepping back. So this is how we design the amplicon. The F1 and R1 are the amplicon pair, and the next pair is F2, R2 as an example. So these amplicons can generate the mini amplicon indicated here. They can generate the super amplicon indicated here, and also can generate the amplicons that we are awaiting, that we intended to design to see. What it actually our technology allows is to avoid the formation of the mini amplicons, and that's how we can multiplex up to 1,000 amplicons in one single tube. By the way, I should highlight that our panel is a one single tube assay in comparison to our competition, where they would split the amplicons across more than one tube. So you're working with just a single tube, so you're not doubling on tubes or tripling. Okay, so just going back to the technology, we would arrange the concentrations of the amplicons to consistently yield the intended amplicons and super amplicons rather than mini amplicons. So that's the core technology, and it's been patented, and it's been run by Swift Biosciences. If you Google patent by Swift Biosciences, you will find it. And then what happens, and this is quite important, if there is a mutation in the amplicon in the primer binding site. So if there is one in the intended amplicon indicated here, in this one here, so it will be trimmed bioinformatically. But if once we are designing the overlapping amplicons, this mutation will be identified and maintained by the primer pair of R1, F1. And in addition, and this is the beauty of our technology, it will be reported in the super amplicon. So you have a doubled confidence that that mutation is present, even though we may trim it with one amplicon pair. Okay. The next slide is, I'm just waiting for the next. Okay. So to give you the another coverage, the coverage of the NY spike mutation indicated here, it's covered by at least one amplicon, and it's been reported in UK and South Africa variants. So indicated here, and then another one screenshot. I wanted to bring up the 69 to 70s spike deletion shown here. And it's been covered, partially covered by this amplicon here. By the way, we don't show the primer sequences here. It's already trimmed. So this is where that amplicon starts, and it's very hard to be able to assay that deletion from that amplicon. Therefore, we have this a second amplicon that would call that deletion. So just to summarize, if there is a mutant, we will able to find it because the panel covers with overlapping amplicon, and at least one amplicon would report for the mutant. And with this in mind, so this is the last slide that we just worked with the company in Portugal called Phylogeny, F-L-O-M-I-C-S, Phylogeny. They developed a suite to analyze our data where you can load your FASTQ files, and it gives you the number of variants. Sorry, these are very small letters here. The number of variants that they would identify, and then they would classify whether this is a UK variant, African variant, Brazil variant, and whatever variant there is reported. So this is a little bit early days, but they're already perfecting the software. So hopefully in the next release, it will be a better graphical output. But what it definitely does, it reports you the variants. So there's a command line already built in. So this is a separate company, Phylogeny, that have this tool, and they are training on their software. They are training the software with our panels. So with this in mind, I just wanted to just go back and highlight the number of panels we have. As I mentioned, SARS-CoV-2 panel with additional coverage indicated here. Then the SARS-CoV-2 S gene. We also cover ACE2 receptor indicated here that I haven't mentioned. So if someone is interested in that as well, it's there. And these are the core kits that I needed to run with each of these panels. And this is the indexing as an example. So unique indexing up to 384 plex. And just to let you know from experience with our customers, with the public health authorities in Norway who are running a lot of our panels in Norway. They're running thousands of samples a month. They find that 384 plex is the maximum that they would multiplex. Otherwise, if you go beyond that, you still have some compromised coverage across some samples, and especially with those that have the high CT values or the low viral load. So with this in mind, thank you very much for your time today, for your attention. I am happy to take questions, and thanks again.
Thank you very much, Gerardus, for a very informative talk. I don't see any questions in the chat. I think everybody is unmuted, so please feel free to raise any questions that you might have to Gerardus.
Absolutely, I would be happy to answer the questions, or you can email to Whitehead Scientific, and they will pass on the. I believe there's one question: is it possible to have the recording? I leave for you, Jacques, to answer.
Yes, so we'll make the link of the recording available to everybody. We have 2 raised hands here. Let me just see. Okay, so Kimberly, I'm gonna unmute you to ask your question.
Hi, I'm a colleague of Kimberly's. We're from the Cellular Lab. I would just like to know from you guys, did you guys confirm those mutations in, and just a recap, how many samples were you working with to begin with? How many samples did you sequence?
When we tested the panel, I think the most extensive study has been done on our panels, has been done on SciLife labs in Stockholm in Sweden, where this was the core facility, where they used 7 different commercially available RNAs with the known mutations. And sorry, not 7, 4 commercially available RNAs, if I correctly remember. And they were, each of them contained 7 specifically introduced synthesized mutations. And they obviously were mixed in the background with universal human reference RNA. And then they used 3 different softwares to identify those 7 mutations. So our panel was able to cover all those mutations and identify, and I will be able to send you the link of that study. So this is an independent study. Swift was not participating in it.
I see,
but they said that.
Sorry, my question is to do with something else. I just want to know that did you guys sort of confirm, because you're using an Illumina platform here, right, to do your sequencing work? Did you then use Sanger sequencing on at least, because according to you, you say that there was a lot of degradation in the RNA, and I mean that makes sense coming from a wastewater facility. So did you guys then at least confirm some of those mutations with Sanger sequencing?
So I understand your point. We are coming from the clinical. So when you look at the gene pan-cancer genes, where you identify the mutation from FFPE samples, for example, and that mutation is probably lower than 3% allelic frequency. So then you would use, and this is a very clinical important sample, and then you will use Sanger sequencing to identify that. But you don't have that much coverage of like 1 million reads per sample. Excuse me. So we haven't done the Sanger sequencing in our lab. But and the authors in the paper from wastewater samples, they haven't validated those mutations. And the reason being that probably they had such a high coverage that there was just no need to validate. On the other hand, these are high throughput labs, so Sanger validation of each mutant would be a tremendous task to do. And
no, I do hear you.
That would be...
Yeah, no, that's not really. I mean, you know, like, but then sort of make a selection of a few that you may just, you know, just to show some sort of consistency, because you know, when you're working with this kind of with this number of reads, it will there's a high, there is a level of errors that can occur, and so just to sort of confirm that yes, it is this mutation, etc. I think it would be good to just take even 10 isolates and maybe try to enrich for them if that's possible, and then perhaps from there just show that, hey, look, it is in fact we are having a low frequency of these mutations at this region of the genome, that sort of thing. So yeah, that's just, I'm just checking if you guys done that. That's all.
No, we haven't done that, and neither the authors in that paper have done that. But yeah, that's a good idea for very low frequency mutant.
Thank you, Johannes.
Thank you.
Lorette, I can leave you to ask your question to Gerardus.
Gerardus, I just want to ask, I think I've got two questions. So the one is just to ask whether the cDNA, before you use it then in your PCR, if you actually do a quantification before you continue with the PCR or with the Swift reagents?
First of all, you would need to do the qPCR on your isolate to see whether there is a viral RNA in there. If that is below 32 cycles, I would expect, so this is a sufficient load, and then you just do cDNA synthesis, and we, in the protocol, you don't quantify cDNA synthesis. You use all the 10 microliters of that yield, what came out of the cDNA, and go straight into the library construction.
Okay,
that's how the protocol is written. Yeah.
Okay. The other one is just with pertaining to the reagents. So you have the various panels, and for all of the panels of primers, you have to use the core kit. Am I correct?
Correct. Yeah, that is correct. So
core
primers do not go separate.
Oh, for all of them, core kit
yes.
reagents are the same. Okay.
That is correct. Yes. So if I go a little bit back to my last slide, so the core kit is universal for all of these panels that you see here, and for the other panels that are gene-specific panels in our offer in our portfolio. Yeah. And just going back to your cDNA, you end up with so little cDNA, almost unmeasurable cDNA, that any qubit readings or any fluorimetric methods would not tell you the cDNA concentration. I spent 7 years in the lab, so I worked with cDNA a lot, not with viral cDNA, but with very low amounts of cDNA. So sometimes you just go straight into the PCR without measuring the cDNA.
You do no enrichment for viral RNA or anything like that, so DNase treatment?
No, because there's just only viral DNA, viral cDNA that we are going to target, and the primers are specific for the viral cDNA or the RNA, okay, if you wish. So you're not amplifying any other sequences that might be in the mix. The primers are not specific to those. So we're not too much worried about the DNase treatment.
Okay, thank you.
Hi, this is Kimberly now. I'm just interested in your overlapping amplicons, where you had the intended amplicons, the super amplicons, and you said you were able to minimize the amount of mini amplicons that were produced. How do you do that?
This is to be honest, I don't know. This is in the part of our patent. And we tend not to talk about this too much, and I have no idea, to be honest. It's our technology that we developed when Swift was set as a company 10 years ago.
Thank you.
Sorry for not being so more explicit.
Boss, would you like to ask your question?
Sure. Yeah.
Lift a little bit on how Normalize works.
Sorry, I did not catch your question. Could you speak up?
How does Normalize work?
Right.
Did
so
you hear
there's
that,
another.
Yeah.
So the normalization principle is as a nucleus protection assay. That's all I can say. So you have introducing a specific sequences that would protect a certain portion of your sample for downstream degradation. For downstream degradation, so you will not degrade a certain portion of your marked sample. That will be carried into the normalized 2 step, where that protection, where the nucleus protection would occur. Yeah. That's how far I can go. Again, this is patented by Swift, and we are going through the patent office patents at the very moment.
Yeah. Okay, thanks.
Radis, we have 2 questions in the chat. One is the 263 SNVs that were detected from water samples, were they of any medical importance? And then the second one that's also joined to that is, what is the acceptable coverage of an SNV for it to be real?
So in the paper, so again, I'm not written the paper, and I just read it as everybody else. 263 SNVs clinically. There was nothing in the discussion that they related to be honest. So I cannot comment too much about that. And what would be the accepted coverage? Most of the SNPs and most of the CNVs are called with 500x coverage or less than 500x coverage, as the authors show in the paper. So I would say anything below 500x would be the most of what you would expect. Then you can go depending on the sequencing reads, and we recommend 250,000 reads, between 250,000 reads per sample to about 500,000 reads. We used to say it 1 million reads, but now we're probably 500,000 reads should be enough. So that would give you, I had it in my calendar, in my notes, that would give you 50,000 X coverage across the genome when you have 500,000 reads per sample or something around those. I just need to double check. I will be able to double check. I don't know I don't remember at the top of my head.
Caroline, if you to ask a question.
Hi, my name is Caroline from Cambridge. So just calling I would like to confirm Caroline. Something. Yeah, so I wanted to confirm at what point do you do quantification? So when you extract your RNA, do you need to quantify your RNA before you proceed to cDNA? That is one. And then at least secondly, from your talk, you've mentioned that we don't need to quantify the cDNA. And then for the library, if you're using normalize in your reaction, do you need to quantify your library, or you just pull after doing the normalize step, you just pull your library and you'll be good to go? Thank you.
Thank you. So going back to the total RNA extraction, I cannot answer that question specifically enough. This is down to the manufacturer of a cDNA that would recommend the input amount of the total RNA. So that total RNA would be in the background of the human total RNA. So I really cannot comment, and I haven't done that experiment, and we haven't done that experiment in-house. So I will refer down to the papers that the customers were using. Now the cDNA synthesis, then when you get after the RT, as I said, we don't quantify that. We use 10 microliters of the end result of the end reaction go into the library construction, into the Amplicon PCR. After you get the library, after the Amplicon PCR and adapter ligation and indexing, at that endpoint, yes, you would need to quantify the library, the yield, which would be, which has to be above 12 nanomolars or 6 nanomolars. Then you go into the normalize. So quantifying with the tape station would give you the molarity. So I would suggest doing that. So doing the tape station runs. After the normalization, yes, the customer has to quantify, because you need to know how many picomolars you need to load on the sequencer again. So the prior normalize and after the normalize of the final pool, the quantitation steps are needed, as it's written in our protocol.
Okay, thank you. And
so that
just
would be my answer. Thank you.
Thank you so much. That's informative. So just a follow-up question is on the quantification bit. If I don't have the tape station, can I just do qubit?
Yes, to a certain extent, yes, you absolutely, you can do qubit if there's no, but qubit would not automatically give you the molarity.
Yeah, you have to calculate. Okay.
Rose, then we have time for two last questions, just for the sake of time. What is the total runtime of the kit from extraction of a swab to the result?
So the all I can say is that cDNAs, they takes about an hour, probably less than an hour, 40 minutes, from what I remember in my lab work long time ago, and then 2 hours for the actual library preparation kit, and then about between 40 minutes to 1 hour the normalization step. So the maximum would be 4 hours. And then you can load into the sequencer and run it sequencing overnight. With our competition, the protocol is 6 hours instead of 4 hours. So some of the customers might not be able to load the sequencer on the same day. So the elegant part is that our protocol is short. The actual library construction kit is time is 2 hours.
Okay. Thanks, Gerardus. And then one final question. Would the panel amplify at least some of the other coronaviruses? Has that been tested?
So the no, we haven't run in the mixture of other coronaviruses. The answer is no, we don't know. We haven't tested it. We specifically selected the primers that would recognize the SARS-CoV-2. But of course, if there is a conserved regions, those would be amplified from other coronaviruses.
Thanks very much, Gerardus, and thanks everybody once again for joining us. As I mentioned, this recording will be shared to everybody, and there will be a short survey that will be sent out to you. Would you please, if you wouldn't mind, just filling that in? And please also feel free to reach out to us if you have any additional questions. And thanks once again for your time. Enjoy the rest of your day.
Thank you too. Bye.
Thank you. Bye-bye. Thank you very much for your time today.